This is a production of Cornell University.
So just to start, I want to orient you a little bit about where I'm from. This is Washington State in the middle there. Washington is a pretty diverse place. It's not just green, rainy Seattle. This is a landscape shaped by volcanoes, glaciers, and ice age floods. So it's really a pretty interesting place to drive around and get out if you like to do that kind of thing. This is where we are in the middle here in Wenatchee. We're about halfway roughly between Seattle and Spokane. Yakima, the Tri-Cities, Hood River below us are kind of the other major centers of the fruit industry in Washington State. And then Portland is down a little bit closer to Hood River there on the bottom. So the primary areas where tree fruit are produced in Washington are pretty close to the Columbia River in this area, this shaded area roughly. And so some of these areas are up the river valleys, and all of the tree fruit in Washington is irrigated. And so the location of where these orchards are is really because of access to water. Ag is a big part of Washington's economy, particularly eastern Washington. The gray counties here are the top 10, as it were. This is, I think, 2010 data from the Washington Department of Agriculture. And apples, for probably 10, 15 years, have been the largest value, farm gate value within the state. They passed wheat and continue to increase. A little bit small print up on the left there, but one thing I want to point out here is that of the about $8 billion in farm gate in 2010, about half of that is horticulture crops within the state. And it's not just fruit, nursery, greenhouse. There's quite a diverse industry. There's bulb production up in Whatcom and Skagit counties as well. But we're going to talk about apples today. This is the Wenatchee Valley. This picture actually I got from Chinook Lee, who some of you know. And Wenatchee on this side of the river, East Wenatchee on this side. This is the Columbia going through town. This is a springtime photo. You can see the lupin and the arrowroot, balsam root blooming here. Everything's really nice and green. Pretty spectacular time of year for us. But as the summer goes on, it's basically a desert where we live, and which has some really big advantages for growing fruit because the humidities are low. There is access to water for irrigation, and so there's really low disease pressure relative to a lot of other places that grow the same crops that are grown there. So it has some advantages in that way. Another thing that's a little bit curious about the Wenatchee area in particular, you'll go back here, is the difference in elevation from the river level up into some of these benches that are around town, which is about roughly about 2,000 meters in difference, which means for something like sweet cherries, you have the same cultivars that are grown within probably a 10-mile radius that get harvested over a month, just because of the difference in bloom dates, because of the elevation. So a little bit of the uniqueness of that. A little bit about apples in Washington. This is obviously the biggest single crop. This year is the biggest volume crop that they've ever had, 120 million boxes, almost 2.5 million tons of fruit, which so far has been relatively easy to market because of what happened to you all last spring. There's quite a bit of nervousness about what happens over the next few years. But you know, a lot of different cultivars. Delicious is still the biggest one by volume, but that's down almost 20% in the last 15, 20 years or so, which makes it actually easier to market everything because there's more diversity people don't get held hostage to all the volume and delicious that the buyers know they have. So that's a lot of apples. And the big reason why there's that many apples, part of it is because there's a world market that can that has the demand, but it's because of post-harvest technology. I think wheat post-harvestites like to claim. And I mean, apple biology lends itself to having the ripening manipulation be ripening extended by storage in controlled atmospheres, particularly low oxygen, the low oxygen component. And so there's installations like this in a lot of places in eastern Washington where you have these just massive rooms that hold anywhere from 800 to 3,000 of these bins, which hold roughly 20 to 25 boxes, 40-pound boxes equivalent of apples. And so a lot of what we do is really related to how can you optimize the system such that you can have maximum flexibility for marketing over an extended period. And so this is really an integral part of being able to deal with this gigantic crop. One of the keys to making CA work is to put the right thing in it. And that's really a key to fruit maturity at the time when it's harvested. I mean, CA is a fantastic technology, but it works best when you have fruit that's at the right stage of development when it goes into storage. And so physiologically, I think those of us that work on apples would say that there's a point in the development where fruit is optimum for its long-term storage, and that's related to the onset of the ethylene climacteric. But from a horticultural perspective and from an industry perspective, that point in development doesn't always match with what the conditions in the field have provided you with any one season. So, for example, this is a series of pictures of Honeycrisp harvested over a number of weeks. And what the trade wants is something over here, right? Because they're red. This is what they can get paid more money for. But there's times when the biology says this is what you should be doing. And so there's this compromise that happens within the industry to balance those two parts of the equation. And early harvest, if the color's right, if the fruit looks good, and it's at a point where the biology is right, great. But that doesn't always happen. So if you end up having to pick later, then there are some issues that the warehouse goes through to determine how long can we hold these things. And for a lot of varieties, that's based on how firm they are. And typically, you're going to have a bit of a shorter storage life as you move across that range of development. Now, I want to talk about a specific example of this here, predicated a bit by the two types of maturity. Physiological, which means that the fruit will ripen after you pick it. It won't just shrivel up and sit there. I mean, apples are climacteric. They go through a specific series of events that makes them get ripe, as opposed to horticultural maturity, which certainly for a red apple is dependent on how red it is. What's it look like? And sometimes these don't match up. And so you're really waiting until you get to what's going to make you money as opposed to what you can store the longest period of time. And for delicious in particular, over the last really 100 years, there's been a selection process where material's been vegetatively selected because of how red it is. And it's being redder being better as far as a marketability standpoint goes. But it's not just redder, it's also when it's getting red. And so if we look at an older cultivar Starkine versus a newer one, Scarlet Spur, the newer ones tend to get redder earlier in the year. And really what that translates into from an internal qualities perspective is that they're redder at an earlier point in development, so that they're harder or more firm at a point where the trade will say these are what we want color-wise. And this is why the strains of Delicious that are produced commercially now are not starking, because if you wait until you get to the color level where you could be profitable, they're too soft. You can't store them. And so over the years, there's virtually none of these produced commercially anymore in the Northwest. It's all these newer strains. So now on to the volatile part of this. So apple volatiles, like it is for other fruit that has aroma as one of its quality components, really are one of the key components to differentiating flavor and aroma among different cultivars. And within a cultivar, this process of generating ripening-related volatile compounds is developmentally regulated. Ethylene is required for this to happen. And so it's very important at the maturity at which the fruit is harvested is really important in how well the fruit can execute this ripening related part of its program. And also the environment in which the fruit is stored has a big impact on the ability of fruit to produce these compounds. And so those are some of the things that I want to talk about for the rest of my presentation here. I'm going to use 3 different examples related to the maturation process, then the impacts of 1-MCP, 1-methylcyclopropene on this process, as well as storage. And then also the last part is going to be some work that we've done relatively recently looking at volatile accumulation during storage rather than after storage when the fruit are taken out and let to sit at ambient conditions. A little bit about the analysis that we use. Typically the work that I've been involved in and that I'm going to talk about today is all based on sampling from intact apples. Part of the reason we've done this is there's kind of been an overriding objective to develop some sort of quality assessment that the trade could use. And seemingly the easiest way to do that for them would be to be able to do this on intact fruit. The other reason is that when you cut apples, or apples in particular, there's a whole nother series of compounds that tend to get produced. And so since we're interested primarily in fresh fruit, we've stayed with this kind of a system for most of the work that we've done. And what we do is we put apples into jars and let them equilibrate for a period of time with a flowing airstream through them. And then we collect the compounds that are coming off the fruit on a absorbent polymer in a glass trap that is then analyzed using GCMS using this system. And this is a pretty efficient process. There are some compromises that are made based on the phase or the material that's used in these traps, but this one, 10X, is actually has a pretty broad spectrum of adsorptivity. And the way that we do quantification based on using standards based on the same system gives us a way to do good quantification of what we get in these samples. So to start with maturity here, a little bit of background. As I mentioned, maturity is really important in determining the capacity of apples to produce ripening-related volatiles after harvest. This is some data from a paper by Song and Banger published in the 90s. And basically what it's showing here, this was with Golden Delicious, is that as harvest occurs later in the season, you go from fruit that's really pretty unable to produce a lot of compounds related to aroma to an increasing capacity as both initially at the point of harvest and then after harvest. A couple of the key things here are that in the first harvest here, where they're getting substantial production, you see that the peak in that production is occurring relatively later than it is in the later harvests. And so what's happened here is that the fruit is matured and then started to ripen on the tree. It starts to have more of a capacity to produce these compounds. But as you pick later and later, the production maximizes and then actually falls off sooner as it compared to the later ones. And so this is one of the things that contributes to post-harvest quality of apple fruit in relation to maturity. Because if you pick earlier, you get a longer storage life, but you have an inherently less capacity to produce a lot of these compounds just because of the point in development at which it's been picked. And so with that as background, we were interested in this problem or this issue specifically in relation to Delicious cultivar. Because Delicious is an apple that gets picked relatively early in its maturation and ripening development because it gets soft. And so if you pick it relatively late, the storage life is compromised quite considerably. And so what we were interested to do was to look at really the seasonal variability in things that are normally measured in relation to maturity, as well as the capacity to produce ripening-related volatiles at the point of harvest, to see how consistent or inconsistent that was on a year-to-year basis. We used this strain, Bisbee Delicious, which is somewhere in the middle between Star King and Scarlet Spur 2. There aren't very many of these around anymore either, because they don't get this red as early in development compared to something like Scarlet Spur. So three of the maturity components that we measured were internal ethylene concentration, starch score with a visual rating using a potassium iodide solution, and then also firmness. From an industry perspective, they don't typically have this information. This is not something that most warehouses generate on their own. So they're relying basically on what's the color here, and then what are these two values telling us about how mature, how ripe are these fruit. If we are going to assume that the fruit is fully mature, physiologically mature, when it reaches an internal ethylene concentration of 1 microliter per liter, and that for long-term CA, the optimal starch is just over 2 on a 1 to 6 scale, and that the firmness is just above 70 newtons, then these are the things that the warehouse would look at the point of receiving and then make their decision as to whether or not this goes into short, medium, long-term storage. I mean, basically, what are we going to do with this fruit to maximize our marketing flexibility? And so in the 4 years of the study that I'm reporting here, there was quite a range, a 3-week range of bloom date. This is the same trees, same block, over 4 different years. And I think there, in all of the years, ethylene started to be produced, starch scores went up, and the fruit got less firm. But when you compare across the years, there's some differences that are relatively important in determining the post-harvest fate, if you will, of these fruit. So, for example, in the first 2 years, the ethylene patterns are relatively similar, a bit higher in the first year. By the last harvest, the starches were up into the higher 2s or above 3. The firmness at the point where the starch was 2 was still above 70. So these are good candidates for long-term CA storage. Before the end, this is one of the things to keep in mind here, is the last 2 points in all these years. When we get into the last 2, you see a couple things is that relatively earlier in the harvest period, the firmness has dropped below 70. And in this year, the starch went up quite rapidly above the point of acceptability for long-term CA, as opposed to last year when it was only at the last harvest when it was that high. But what would happen here, if you wanted to, say, pick in this year for long-term CA, you're going to have to pick pretty early. Starch was going to be low, and likely these fruit were not going to be red enough. And so basically what would have happened is they would have had to pick later and not put them into long-term CA. So the analysis of the volatiles that were being produced, and this is not including ethylene. This is the other things that the fruit are producing. A principal components analysis here that accounted for just under 70% of the variability. You see that we've got nice patterns related to the 4 years of the study. The red one is the late bloom year, which will make more sense when we get a little bit farther on here in the presentation. But one of the things I want to point out is that in all of the years, the data is pretty well clustered around the same points early in the development period. And one of the real differentiating factors between the years is as you get closer to the later harvest dates, where things are a little bit more different than they are earlier. And the loadings plot for the same data, really this is driven by the onset of production of a lot of ripening-related esters that tend to cluster in the top of this plot. The things down in the lower areas tend to be alcohols, aldehydes. There's a few esters in there. But really once maturity, physiological maturity has been attained and ripening starts and the fruit starts making esters, that's really what separates out the data set. So this is all of the things we measured here. Same arrangement, same bloom dates across this way. The plots at the 5 things at the top here are growing degree days. This is starch, ethylene, hours below 10, and then the incidence of, or the severity of superficial scald after 6 months in storage. Because scald is related to maturity at harvest in most years. And so what you see as you look across this plot is that in all of the years, the biggest change in volatile production was at the last 2 harvest dates. And it was really relatively abrupt, where not much was happening before we got until these last 2 harvest dates. In the year where the harvest or the bloom was relatively later, there was quite a different profile in a number of compounds which tend to be alcohols, aldehydes, which serve as precursors for ester production. I don't have an explanation for why this happened, other than this happened in a year where the fruit really had a relatively less days on the tree, and it was also its initial stages of development was during a period of year where the environmental conditions were somewhat different than they would have been when bloom was earlier. I mean, these are components of, say, yearly variability in production of these things that aren't really very well understood, because it's difficult to do this kind of research. But so I guess one of the messages here is that these patterns are not consistent from year to year in some aspects. Even the last 2 harvest dates being the point at which there was the biggest change in volatile production, if you think back to the previous slide, there was the other things that we were measuring, ethylene production, starch, and firmness, didn't necessarily follow that same consistent pattern. And so what's happened here is a couple things. Oh, let me step back here a second. The group within this cluster, which you can't really see it as well on here as I can see it here, tends to have less of a change over this period of time compared to the bottom ones. These are a group of ethyl esters which, as you let delicious fruits sit out in the bench and get ripe, these tend to increase a bit later than some of the other compounds do. And those being the group in the bottom 2 clusters here, which tend to be have a different alcohol moiety in the ester: butyl, hexyl, propyl, pentyl, and then 2-methyl-butyl acetate. And these compounds you can make, if you take a bunch of these and make a mix of the pure standards, you can make something that smells pretty much like a nice, ripe, red, delicious apple. So these tend to be produced in volume relatively earlier in the ripening process than the ethyl group does as a comparison. So what that said to us is that, like the other components of maturity that we measure, this process is a little bit different every year as well. And when you think about how this happened over the 4 years in relation to the other components of physiological and horticultural maturity, this increase came after the point of horticultural maturity in some years and before it in others. So when it comes after, what that means is that this fruit is off the tree before it reaches this stage, and it's not going to have the capacity to produce a lot of these things, meaning that they're going to taste different, even though they may look the same as they did the year before. And it's one of the things that makes delicious have some people question that part of its name. So next, this is the impacts of 1-MCP, really the influence of ethylene on this process, as well as part of what storage does to the capacity of fruit to produce these compounds. So for this part of the study, we used Gala. They were treated with 1-MCP, very amazing inhibitor of ethylene action, highly used in the commercial trade around the world anymore. And then they were stored either in air or in a controlled atmosphere at 1 kilopascal oxygen and 2 CO2, which is pretty standard for the industry in the Pacific Northwest. So these fruit were held in storage, and then we took them out and let them sit for a week at 20 degrees, which is kind of a standard simulation of transit and retail for apples after they come out of storage, to see how it would impact components of quality, including production of volatiles. So there were a number of treatments. This is actually kind of old data when we were trying to look at how much MCP is enough, as it were. And so we went from 0.1 to 10 microliters per liter of 1-MCP at harvest, and then stored in air or in CA alone, or with a treatment of MCP prior to going into storage. Relatively early in the storage period, taken out every month for 7 months, you see this differential. Really, even at 1 month, there's a difference relatable to the fruit having been treated with 1-MCP prior to storage. And as the storage periods extends, basically these things really aren't changing very much. The blue circles and the purple ones are the fruit that have been stored in CA. After about 4 months, the group treated with MCP but then stored in air starts to change a bit and does differentiate between the CA, but still is higher than the untreated fruit and the lowest MCP treatment. And it's similar for titratable acidity, which is another component of quality that's important for flavor and aroma. Not aroma, but certainly for flavor. So a loadings plot of the volatile results from this. And you see on the left, the treatments progress up and out in the plot with the concentration of MCP. And then at the top, the combination treatment, MCP, CA, or CA alone. And what tracks over here is the bottom group here, again, is primarily acyl esters that really give the fruity components, the ones that are produced relatively earlier in ripening for Gala. And as you go up and then into the CA area, these, again, there's a lot of these ethyl group ones that are present up here. And really what this is showing is that these tend to be made much more in volume related to these treatments than to the MCP or the CA ones. So esters alone over the same period, the controls in the lowest MCP treatment, which didn't do much for firmness or acid, you see we had this initial increase and then another increase later in storage. The later increase was also happened in the fruit that had been treated with the higher rates of 1 MCP. Either group stored in CA stayed relatively low at the longest storage durations compared to the other MCP treatments. So this biphasic pattern, if you will, is a bit unusual. And when we went in to look at what individual compounds were doing in relation to this pattern, you see that in the initial period for the controls and the lowest rate of 1 MCP, that there's production of a number of compounds that is higher than it is in the treated fruit. And what these are, again, is this series of relatively these simple acyl esters that don't include the ethyl ones. And so then when you look at the 5 to 7 month period where we started to see an increase in a lot of these other compounds within the checks and the low MCP treatment, we also saw that in all of the MCP treated fruit. But we didn't see this group alone once the MCP treated fruit started to have the capacity to produce these compounds. And so what appears to have happened here is that the treatment with 1 MCP didn't just delay the onset or the production of a lot of these compounds. It really changed the pattern of how they were produced, such that you miss this initial period where you're really only getting these real fruity, more characteristic group of compounds, such that when the fruit regains the capacity to produce these, it's a different suite of compounds, if you will, compared to this initial period. And so, why this is happening specifically is something that remains to be determined. But it's not a simple, well, the fruit is old, it's starting to get senescent or overripe, however you want to describe it, because these fruit could be classified that way. They're soft, they're low acid, but these aren't. I mean, these are still firm, they still have a lot of acid. And so what it means is that this whole process has been altered by having ethylene action removed from the equation, as it were, for a period of time, such that it doesn't really recover the capacity to ripen normally. I mean, they're getting ripe and they're doing things that gallows do during that process, but it's not the same pattern that you see in the controls. Yeah, again, when the second production is this ethyl group again, along with a number of aldehydes. So it's a different group of compounds as opposed to the one on the bottom. So I think what this illustrates is, MCP is having a big effect on this. In the trade, gallows aren't stored for a period of time such that they would experience this increase in air, because if you're going to store them for a long period, they're going to be this. They're going to be the ones that are in CA. So what does that mean about what they're going to, what are they going to taste like when you put them in your mouth? And it turns out that's not as simple of a question as you might think it is. This is some work that Gene Kupperman, colleague of ours at WSU, and Anna Marin from Oregon State University really were the drivers behind this a few years ago. And their question they wanted to ask is, how do people perceive 1 MCP treated fruit versus non-treated fruit that have been where you've selected using a non-destructive firmness technique, so that firmness is pretty much the same. It's not like you're taking controls that haven't been treated that are soft and comparing them with treated fruit that are still hard. These are all about the same firmness. We did the volatile analysis for a number of key compounds, and I hope you can see this, that the bold columns on the right, which are the CA or fruit stored in air that haven't been treated, are all significantly higher than the fruit that have been treated with 1 MCP and then stored either in air or in CA. But what the folks they gave these to test, and they basically said, do you have a preference, and what is your preference, was pretty interesting to me, in that when you look at the mean liking scores for all of these groups, MCP air versus control CA, MCP CA, control CA, or MCP CA versus new crop fruit from Chile that hadn't been stored, these scores are all pretty similar. And the preference scores in general favor the fruit that have been treated with 1 MCP. So what does that mean? It means that people's preferences for apple flavor are different. You know, it's not just a matter of saying, you've got a lot of these, they're going to be better. Well, some people that may be why they thought these were better, but it's not that simple. And so there's been a lot of work like this done trying to sort this out with MCP on apples and other crops. And it really, I think, depends on the questions that you're asking. I mean, if you want to say that MCP makes something better or worse, you can do that by the way you set up these consumer type tests, or even taste panel with trained panels. So it's not as simple of a question as more is better. And I think the other part of that is that if you think about the differential in production of different groups of esters as ripening progresses, as the fruit starts to get older and into the phase where it's producing a lot of ethyl esters, they don't taste the same. I mean, that changes how the fruit tastes. When you combine that with softening, more may not be better in that instance as far as how that impacts quality. So on to the last part, and this is volatile production during CA storage. It turns out that there's not a lot of literature about what happens while fruit is stored in low oxygen or high CO2 controlled atmospheres. Most of the work like this has been done, as I described earlier, where fruit is taken out, it's let sit for a while to simulate shelf life, as it were, and then you do the analysis. So we've been interested the last few years to try to develop information that provides additional management tools, as it were, to industry to make determinations about fruit quality and marketability while it's still in storage. And so one of the things that we started to do was to set up experiments where we would actually do the volatile measurements while the fruit was still in CA. And for this group of stuff, we picked 2 situations where the environments were set up so that they would cause problems. The first one being low oxygen, where we went down to 0.3 kilopascals oxygen to try to generate low oxygen injury. The reason you would actually try to operate that way commercially is to try to prevent this disorder, superficial scald, which low oxygen can be a way to at least delay. And the other one is high CO2 injury of Fuji, which develops this really wonderful internal browning with cavities that when you look at the fruit, you can't see that it has it. So the way we collected the samples for this is we have our chambers, our research scale chambers. They hold about 8 of these fiber trays of fruit. And same collection system. Here's our 10x trap, a little pump where we would circulate the atmosphere from these chambers onto the traps, and then we could basically do the analysis on the traps the same as I described earlier. And that way we don't have to take the fruit out. We're not changing the atmosphere. It's staying under the low oxygen or high CO2 conditions while we're doing the sampling. This is the ports on the side of the chambers to get the gas in and out. So for the delicious part of this, I just want to briefly show you, we've done this for 3 years now, and the first 2 years of this, the fruit was relatively similar starch-wise, a bit higher in ethylene the first year. All 3 years, the firmness was pretty consistent. The last year, the fruit had much higher ethylene. The starch scores were higher. The acidity was lower. So these are fully mature and started to ripen, I think, a bit more than the first 2 years are. All from the same orchard, managed the same. So the low oxygen concentration on the left, the amount of time in storage on the right, and these are not equivalent periods as you go down the axis here. They're weighted towards the front end such that these are all about a week apart, and then it starts to get longer. So what we saw relatively early on in the lowest oxygen concentration was an accumulation of compounds that we weren't seeing as we increased the oxygen or as we were storing fruit in air. And guess what these were? Again, it's this whole series of ethyl esters, including ethanol. So not surprisingly, apples make esters really largely based on the alcohols that are present. So if you have a lot of ethanol there, you tend to get these ethyl esters earlier than you would if, in this case, because these apples are not ripe 2 weeks after harvest. Then there's other groups where the changes are not as affected, really, other than the CA compared to the air fruit. And then there's a whole group down here which are, again, a lot of the early ripening related compounds that are being produced much more in the fruit stored in air than they are in the ones in any of the CA environments. And really, one of the things that, regardless of low oxygen concentration, these tend to be produced less, which I think, if you eat fruit that comes out of CA, you inherently know that. There's a lot less production, or you just don't taste these as much as you would if you're eating a ripe one that's never been in that same condition. So low oxygen has this effect, as it does with galas. The different atmospheres did a predictable job of managing firmness and acidity. The lower letters here are the differences within months. So the CAs are higher. All of the CA treatments are higher than the fruit stored in air. It was only at 8 months where the lowest oxygen in this experiment was higher than the other two. So, again, we're restricting part of the ripening process here by these treatments, but the differential in oxygen is having a pretty big impact on what the fruit are making. So one of our objectives here is, okay, can we use measurement of a fewer of these to provide some indication that there's an issue or not an issue? And quantitatively, as you might expect, ethanol, ethyl acetate are the compounds being produced the most. This is in 1 year of this study. All of this data, I'm sorry, was from 2011. And so if you're going to try to use 1 or 2 values or compounds to try to make this assessment, you either need to have a pattern that's different consistently, or you need to have a threshold value above which you would say there's a problem. So here's the lines, the slope of the line of accumulation in the first week. Clearly much more rapid in the lowest oxygen than the fruit stored in air or the other oxygens. But if we look at the amounts, in this year, the only treatment that got above 10 nanomoles per liter was the lowest oxygen. And for ethyl acetate, it was somewhere between 4 and 6. So maybe that's a range where if you were going to try to do this commercially, you could say this is what we want. Well, we've done this for 3 years, and in 2 of the years, the patterns have been relatively similar. These 2, where we saw this initial rapid or relatively rapid rise, and it continued to go up for within the first 3 months. The kicker is this one, where this fruit at 0.2 was anaerobic. It had plenty of ethanol, but it didn't accumulate very well until almost 3 months in storage. And so if you're using, we have to use, if we're using 10, we're not going to know there's a problem until we get out to here. And that's going to maybe be too late to do something about it within the room. That's what this line is in this year. Once one of these groups that was stored at 0.3 got up above 10, we raised the oxygen to 1.2, and you see that over time, both for ethanol and for ethyl acetate, well, not as well here, the values start to go back down, meaning that the fruit can recover once you put enough oxygen back into the room. The other issue here is that as opposed to the other year when we used 0.3, where 0.8% never induced enough ethanol to get up above 10, it did in this year. So I think, this is only 3 seasons with 1 lot. So when you think about this in relation to a whole room full of fruit from different lots, setting a threshold value to try to do this might be more difficult than you would think as you think about the biology of fruit and why this happens in the first place. So I think that was one of the messages or the lessons that we got out of the stuff from Delicious. For Fuji, only the fruit stored at 5 kilopascal CO2 got CO2 injury. Okay, so there's a large group here where relatively little difference between the treatments. And this is the fruity stuff that tends to get produced relatively early. There's no ethyl esters in there. When we look into this cluster, ethyl esters and some methyl esters as well. So even at high CO2 is causing a differential production of a bunch of these compounds as opposed to the fruit that are stored in air. I think one of the issues here, again, okay, what could we pick that might tell us there's a problem, is a bit more difficult because the quantitative relationships between these treatments aren't really very different. Here's ethanol over here, and you see that this doesn't increase to its maximum until pretty far. This is 6 months into storage here. And so maybe not surprisingly, we didn't see that happen. But when you look at what production of a few of these did, some of them, ethyl butyrate, methyl butyrate, early on there was an increase in all of these. But the treatment differential, 5 to 1.5 to a half, that's not a big enough difference, I think. If this was something you would see consistently to be able to say this much is telling you have a problem. And what we see over with ethanol is that Fujis just inherently make ethanol pretty well. And so I think what happens early on is they can pretty efficiently take this and make it into esters. And so we're not seeing as much of an accumulation early as we do late when this capacity to produce ethanol continues to be there, whereas the capacity to produce esters with it actually decreases the longer the fruit's been in CA. So that's probably part of the reason why we're getting this accumulation later in storage. So I think that's where I want to stop. And I guess the things that we've taken home ourselves over the years is that harvest date, season are really important. And it's important in ways differently for different cultivars just because of the way different cultivars mature and ripen before they're ready to be picked. Ethylene action is critical to this process. Controlled atmosphere has effects even within the different gases manipulations that we do. And really it comes down to biology for what the fruit's going to do, and the business having to make decisions based on what the biology has already done. And I think that's the challenge for the industry in what they do every year. So these are the folks that did all this work. Dave and Janie are technicians. Luis and Jutong were postdocs. Christy is a master's student who just finished, and Dave Riddell is one of our colleagues in the lab. So thank you. Right. For the MCP, actually for the controls, it never really dipped as much as the ester production did. It was a really, I mean, I think that's the only time I've seen that biphasic pattern. And so we've done some, we're actually repeating this again this year to see if we get that. So there wasn't as much of a change, but in the MCP treated fruit stored in air, that's when we saw ethylene start to be produced again. Yeah. Gosh, I'd have to go back and look at that paper. There's a lot more in that paper than that table, but they were very careful to try to minimize the impact of firmness here. And this acoustical method for doing this, I think a lot of people would argue is, I mean, it's you have to use that value with a little bit of understanding that it's measuring something, but if you get groups that have the same value, I think my personal opinion is that you can still tell differences because the texture isn't necessarily the same between those. And so that's part of what people were looking at here. And this was consumers. These were people at the Portland Sunday or the Portland Farmers Market is where they went and did this. So it's not trained panelists. It's just people who like apples. People, I think, that plot is from people who eat a lot of galas, in fact. So even within that population, there were people that said, we like these, even though it's not making these other compounds. I think that my experience certainly has been that MCP and or CA really don't impact sugar as much as they do other parts of ripening. And that it's really determined at the point of harvest how much starch is there that's going to get broken down. I mean, ethylene action doesn't appear to have much of an effect. Once the starch degradation process has started, it pretty much just goes. And so there's less of an impact on sugar as opposed to a lot of the other things. And so one of the reasons that I have acidity on there for the gala example is that before MCP was ever around, one thing that we had noticed is that because in part of the impact that maturity assessment for Red Delicious and its offset from the capacity to produce volatiles, putting that into long-term CA, where even if they could produce volatiles at harvest, it's not going to be very efficient after they come out of CA, that acidity becomes a relatively more important component of quality. Because when it gets too low, and that number is different depending on the cultivar, the fruit just don't have much going for them. And so you better have acid left at the end because you don't have much of these other things. And I think when you look at the changes in sugar as opposed to harvest, it actually goes up a little bit because starch is being metabolized. But as a percentage change, the acidity change is considerably higher than it is for sugar as fruit ripens and it sits in storage. There is, and it's different for different cultivars, in part because not everything, inhibiting ethylene action isn't 100% positive for all cultivars. We can argue that it's not positive for aroma production. But it also, in some cultivars that are sensitive to CO2, it actually extends the period over which they can have issues if they're exposed to CO2. And so for Fuji, for example, where we're from, you don't want to treat them right away when they're harvested, because it makes it more likely that you're going to have CO2 injury, as opposed to something like Gala or Delicious, which where firmness management is much more of an issue. So you want to treat them as soon as you can after harvest, because then it has the maximum impact on preserving firmness and minimizing firmness loss. The longer you wait, the less of an effect typically that you see. But one of the first experiments that we ever did was in March with Fujis that we got from a warehouse. And we treated them and then we let them sit out, and we could detect differences even that late in the year. I mean, it wasn't something that would have been commercially significant, but ethylene is important throughout that process, at least for some components. And so if you interrupt the ethylene activity, there are things that happen even relatively late, in our example with Fuji's. So 1-MCP is a gas, for those of you who don't know, and so the current license holder has developed a formulation that can be field applied, which seems is a kind of a technological achievement, at least and whether or not there are impacts related to disorders from pre-harvest treatments, I'm not sure that that's been very well characterized. I mean, I haven't been involved in a lot of that work. I don't know, have you guys seen anything related to Harvista and disorders? Actually, I take that back. Jennifer Diel has seen, and actually we've seen this too now I think about it, that some of the Honeycrisp disorders can actually be less if the fruit has been treated with MCP in the field. And it tends to be those that are, they're chilling disorders, but they're related to maturity. And so I think what's happening is that when you treat them in the field, it's slowing down the maturation. And so it's prolonging the period where they're not as sensitive to low temperature. I think there's a lot more work that needs to be done in that area. But because right now, one of the things that the commercial business is trying to do is to minimize the post-harvest effect of this material, because they don't want to jeopardize their post-harvest business. But I think there's a lot of opportunities for that for different reasons other than just disorder management that remains to be seen at this point.
This has been a production of Cornell University, on the web at cornell.edu.